Assalamu alaikum, second year. Welcome to welcome back to online learning. Today we will start with the chemical senses, which are olfaction, that is the sense of smell, and gustation, which is sense of taste. But first, let me ask an interesting question that why your sense of smell is reduced when you have a cold, even though the cold virus it doesn't directly adversely affect the olfactory receptor cells. Well, you think over it, and the answer will come along as we move on with this lecture. But first, I give you a brief introduction to the chemical senses as why they are termed so. The chemical senses they provide a quality control checkpoint for the substances available for ingestion. These chemical senses they are also classified as visceral senses because of their close association with gastrointestinal function, including the flow of digestive juices and their effect on our appetite. Well, as far as the reception of these chemical senses is concerned, the receptors for both smell and taste they are called chemoreceptors because they generate the neural signals on binding with particular chemicals. Now, the same receptors they are also classified as exteroceptors because the stimuli arrive from an external source, and stimulation of these receptors induces pleasurable or objectionable sensations and signals the presence of either something to seek, which is nutritionally useful and good tasting food, or they can signal the presence of something to avoid, which is potentially toxic or a bad tasting substance. Now, I tell you a few interesting bits about the developmental aspect of our chemical senses. You see, the sense of smell, it doesn't have a chance to develop until after birth. But by 10 days old, a baby can distinguishes the scent of his or her mother from another person. Similarly, the sense of taste also develops rapidly. Even 2-week-old babies, they prefer sweet tastes over others. Since babies learn by putting things in their mouth, it is important to watch them closely for this reason. Enough with the introduction, and let's start with olfaction, which unfortunately is poorly developed in humans. But still, human olfaction is capable of distinguishing between roughly 10,000 unique odors. Interestingly, sense of smell can trigger memory as the smell analyzing region of brain is closely connected to amygdala and hippocampus that handle memory and emotion. Now, we can categorize animals, including human, into three groups based on their olfactory powers. Animals with the greatly developed sense of smell, they are called macrosmatic, while animals with the less developed senses, they are microsmatic. For example, humans and monkeys. And then there are animals which have no sense of smell and they are called anosmatic. Many aquatic animals, they are the example of anosmatic animals. Now, as far as the importance of olfaction is concerned, I myself am quite obsessed about it, but for you ordinary beings, just know that it is important for the enjoyment and selection of food. Moreover, the flavors which you so often crave, they are a combination of taste and smell, where smell contributes about 80%. More crucial is that olfaction gives warning of harmful substances or places. It even becomes life saving in lower animals, where smell also plays a major role, not only in finding direction for seeking prey or avoiding predators, but also forms a part of sexual attraction to a mate. Keep in mind that to be smelled, a substance must be sufficiently volatile for entry in nose with inspired air, which means that it is easily vaporized. Then it should be sufficiently water soluble to be dissolved in mucus, and finally, the substance ought to be at least slightly lipid soluble, as lipid constituents of cilium itself, they are a weak barrier to non-lipid soluble odorants. Now let's move on to the basic structural functional unit of olfaction, which is its membrane or mucosa. You may find it interesting that the olfactory epithelium is one of the few regions of body where central nervous system nerves they directly interface with the external environment. The membrane is located in the upper part of the nasal cavity with an area of around 2.5 to 3 square centimeter, and it is inhabited by three types of cells. First, there are olfactory receptor cells, which are replaced every 2 months, and they also decline with age, which would mean that approximately 1% of these sensory receptor cells they are not replaced each year. Next are the sporting cells, which secrete mucus, and finally there are basal cells, which serve as the precursors for new receptor cells. And do not forget that mucus is present on top of our olfactory membrane. For the reception of odorants, there are olfactory receptor cells, which are bipolar neurons derived from our central nervous system. Their number is about 100 million and they are of 1,000 different types in each individual. Keep in mind that a given receptor can respond to a particular odor component. Although there are millions of olfactory sensory neurons, each expresses only one of 500 olfactory genes. Now, the receptor cells are interspersed by much smaller number of sustentacular cells. And then there are basal cells along the basement membrane of the olfactory epithelium, which regularly divide and yield differentiated cells that replace lost neurons. It's very important to understand the working of receptor cells. So, let's dig a little deep for the olfactory receptor cell. You see, it's a bipolar neuron that have an average lifespan of 48 days and then it is replaced. Apical surface of receptor cell exhibits a knob that emits 4 to 25 olfactory hair or cilia. These cilia are non-myelinated with a length of 2 micron and a diameter of only zero point 0.1 micron. Cilia are important because they contain the receptors which provide binding sites and project into the mucus. Axons of the same olfactory receptor cells collectively form the olfactory nerve. So, you see, now you can see that how important the reception is always, and so are the receptor cells. Now, what are these? These glands are spaced among the receptor cells to secrete mucus onto the epithelial surface of the olfactory membrane. So far, I have mentioned about the olfactory mucus couple of times. So why don't we just obtain a teeny tiny bit more of knowledge about it? This olfactory mucus contains orden binding proteins that help ferry the hydrophobic odorants to the olfactory receptors. Mucus also contains some proteins which increase the actions of odoriferous substances on receptor cells, and then it contains lactoferrin, lysozyme, and various immunoglobulins that help ensure that pathogens do not gain access to central nervous system via the olfactory nerves. Next up is the olfactory bulb. Here in this figure you can see the olfactory bulb, and this structure is magnified here. It's a neural structure of forebrain which is involved in olfaction, and here you can see that it lies over the cribriform plate of the ethmoid bone, which separates the cranial and the nasal cavities. Well, you already know about the olfactory receptor cell. Now, let's see how this receptor protein plays its role in the working of receptor cell for olfaction. This protein is located in the membrane of each olfactory cilium, and each receptor protein is a long molecule which threads its way through the membrane about 7 times, folding inward and outward. Now, among these folds, outside fold binds with the odorant, while the inside fold is coupled to a G protein. This G protein itself, it's a combination of three subunits, alpha, beta, and gamma. Now these subunits, they have their own importance, which I will tell you shortly. Interestingly, individual olfactory neurons express a single receptor gene only, because the number of the odorants that a person can distinguish exceeds a receptor gene number by several orders of magnitude. It means that the receptor must recognize specific chemical groups of multiple odorant molecules rather than responding to just one odorant. Let's look at how an olfactory cell is excited. Look at this figure, and you can see that first of all, orden molecules diffuse into the mucus. Then they bind to the receptor protein that is linked to a cytoplasmic G protein. And on binding of the odorant, the alpha subunit of the G protein, it separates away. Now this separated unit, it activates the adenyl cyclase when it binds here, and this results in the formation of cyclic AMP. Now this cyclic AMP, that is responsible for the opening of cation channels through which the sodium ions, they diffuse inside the receptor cell and depolarize the cell. So this is how the receptor cell, it is excited. Upon stimulation of the olfactory cells, the depolarization of receptor cell leads to production of action potential in the olfactory sensory fibers. A membrane potential of an unstimulated olfactory cell is minus 55 millivolt with baseline activity of action potentials once every 20 seconds to 2 to 3 per second. But the depolarization, it brings the membrane potential to minus 30 millivolts with a frequency of 20 to 30 action potentials per second. So you can see that this is how the olfactory transduction is achieved. As you can see in this figure, that olfactory transduction process, it can be summed up in three steps. Number one, the orden molecules, they bind to the receptor and activate the G olfactory protein. And then next, the cyclic AMP, it causes calcium and the sodium influx. And finally, the calcium ions, they open a chloride channel, and the olfactory receptor neuron spikes, resulting in depolarization. Now, this cascading effect, which is seen in the olfaction, it basically is a pattern to enhance the effect of a weak ardent molecule. And now we will see that how is it achieved. Actually, what happens that a single dissolved molecule, it can activate many receptor proteins, and activated G protein complex activates multiple molecules of adenylyl cyclase. Now, each of these molecules causes formation of many times more molecules of cyclic AMP, and each cyclic AMP opens still many times more sodium ion channels. So you can see that this process multiplies the excitatory effect of even the weakest order, and greatly enhances the sensitivity of system to even a slightest stimulus. Although the determination of differences in the intensity of any given odor is poor in olfactory system, the intensity of initial olfactory stimulation is proportional to the logarithm of the stimulus strength. Now, the concentration of an odor, it must be changed by 30% before a difference can be detected. Well, you might have noticed that our olfactory sensations show rapid adaptation. To achieve this rapidity, our olfactory receptors adapt about 50% during the first second, but thereafter they adapt very little and very slowly. Now, this olfactory adaptation is mainly a central mechanism which is achieved through the adaptation of receptors, because the olfactory receptors, they are phasic receptors, and also through the psychological adaptation, which as a matter of fact is far greater than the receptors adaptation. But don't forget that additional adaptation also occurs within central nervous system. Now let's see how this olfactory adaptation is centrally regulated. Neural mechanism for adaptation involves centrifugal fibers from brain backward to the granule cells. Now keep in mind that these granule cells, they are the inhibitory cells in the olfactory bulb. So, this feedback inhibition suppresses the relay of the smell signals, providing adaptation. And you can also infer now that it's not a physiological process which takes place at the level of receptors, but rather it's a mechanism which alters perception. Primary olfactory sensations are as many as 100, but they have been narrowed down to seven, which are camphoraceous, musky, floral, peppermint, ethereal, pungent, and putrid. Olfactory thresholds are the lowest concentrations of a chemical that can be detected. We all discussed that even a minute quantity of stimulating agent in the air, it can elicit a smell sensation. For example, there is a chemical methyl mercaptan which can be smelled even when 125 trillionth of a gram is present in each ml of air. Now, here you can see the olfactory thresholds of some common odors. And here are some other examples of the substances which may be detected at very low concentrations. But beware, because there are some toxic substances which are odorless. It means they have odor detection threshold higher than their lethal concentration. One such example is carbon dioxide, which is detected at 74,000, but the lethal limit is already reached at 50,000. Well, that sounds quite dangerous, I must say. Now, if I want to sum the whole debate so far, I can say that smell sensation either could be pleasant or unpleasant, and threshold of some ordered molecule is extremely low. for example, 125 billionth of a milligram, but the range of sensitivity is only 10 to 50 times. Now, I don't want to skip an important thing as orden binding proteins. You see, our olfactory epithelium contains one or more orden binding proteins, which are produced by sporing cells and then they are released in the extracellular space. Now, these orden binding proteins they have certain functions. These proteins may concentrate the ordens and transfer them to receptors, and they may also partition hydrophobic ligands from air to an aqueous phase. And then they sequester the ordens away from site of the odor recognition to allow for oral clearance. Now we jump to the olfactory pathway, which is responsible for the transmission of signals into the central nervous system. The olfactory fibers, which are axons of receptor cells, collect into bundles of 20 or more, and then they pass through the perforations in the cribriform plate of a ethmoid and finally enter the olfactory bulb. You see, olfactory bulb is a complex neural structure containing several different layers of cells, and each olfactory bulb is lined by small ball-like neural junctions which are called glomeruli. So we can say that fibers terminate in relation to glomeruli. Olfactory glomerulus actually forms the first relay station. This is a tangled knot of mitral and tufted cell dendrites and olfactory nerve fibers. Now, each of the glomeruli receive synaptic input from only one type of olfactory receptor, which in turn responds to only one discrete component of an organ. Then the glomeruli sort and file various components of odoriferous molecules before relaying signals to the higher levels. Keep in mind that the mitral cells in glomeruli they refine the smell signals. Now these olfactory glomeruli they also have some other functions. For example, they demonstrate lateral inhibition, which sharpens and focuses the olfactory signals. And this mechanism is mediated by periglomerular cells and granule cells. Moreover, extracellular field potential in each glomerulus oscillates and helps to focus the signals reaching the cortex, while the granule cells they regulate the frequency of oscillation. Then comes the olfactory tract, which is formed by the axons of mitral and tufted cells. It leaves the olfactory bulb after receiving signals and enter specialized regions of the cortex. Here you can see that both olfactory tract and bulb they are an anterior outgrowth of brain tissue from the base of the brain. A unique feature of our olfactory tract is that the tract does not first pass through the thalamus before reaching cortex, while all neuronal communication involving sight, hearing, taste, and touch, they make a mandatory stopover at the brain's relay station, the thalamus. It means there are no direct flights between the sense and the cortex except for smell. The cortical areas of olfaction are medial and lateral. Medial olfactory area exerts primitive behavioral aspects of olfactory signals. For example, licking, salivation, and other feeding responses caused by smell of food or by emotional drive associated with smell. And this area is represented by septal nuclei. Now, signals from this area they project to hypothalamus and other regions for controlling the same aspects of olfaction. While the lateral olfactory area is concerned with the specific behavioral responses related to odors, such as a learned control of food intake. One example of the affective domain of this area is the aversion to food that has caused nausea and vomiting in past. Now, the lateral area itself is composed of piriform area, and also cortical amygdaloid region. From these areas, these signals they are directed to less primitive limbic structures, for example, hippocampus. And when we continue with the olfactory pathway, main olfactory destinations they are primary olfactory cortex, including the piriform cortex, and then there is amygdala and the entorhinal cortex. In us humans, signals from primary cortical olfactory area they are projected also to the dorsomedial thalamic nucleus and then to the orbitofrontal cortex. This route is phylogenetically a newer pathway, and it is involved in conscious perception, also in the analysis of order, and at the same time also in order discrimination. Now the question is that how different orders are discriminated from one another, but this matter is exactly not resolved. Although there is a theory that receptors are selectively sensitive. For example, if two orders are mixed, the resulting intensity is always less than the sum, and perceived intensity is dominated by stronger component due to the receptor action. Then there is a direction from which an order comes, which may be indicated by slight difference in the time of arrival of orden molecules in the two nostrils, just like we did it with sound. Let me know if you guys are familiar with the term of pheromones, which I'm pretty sure you are. You see, the pheromones are non volatile, orderless chemical signals which are passed subconsciously from one individual to another. And then there is a vomeronasal organ, which is abbreviated as VNO. It's an accessory olfactory organ found in many animals, including mammals. It is located half an inch inside the nose, next to the vomer bone. Now, you must be wondering that what's the connection of pheromones with VNO? Well, I will enlighten you by telling you that the other name of these famous pheromones is vomeropharynx. In humans, vomeronasal organ was considered as vestigial or non-functional. But recently, it is found that this organ is present in the form of vomeronasal pits on anterior part of nasal septum. And receptor of these pits, they detect odorless human pheromones even at a very low concentration in air. And why this organ is also called Jacobson's organ? Of course, because this was discovered by Ludwig Jacobson in 1813. Now, tell me that do we have a sixth sense or for you a common sense? Well, let's discuss. You see that binding of a pheromone to its receptor on the surface of the neuron in VNO that triggers an action potential. And this action potential travels through non olfactory pathways to the limbic system, and hence it governs the emotional response. It is interesting to note that the messages which are conveyed by VNO, they bypass the cortical consciousness. So this subconscious detection of odorless chemical messengers in the air, that is considered as an extra sense for humans. Now we will look at the abnormalities of olfactory system. The first one is anosmia, which is total loss of all the odors. Now, anosmia could be temporary or permanent. The temporary anosmia is usually due to obstruction of nose, which occurs during common colds, nasal sinusitis, and even in allergic conditions. While the permanent anosmia, it could occur during lesions in olfactory tract or meningitis, and also in some neurodegenerative conditions such as Parkinson's disease and Alzheimer's. Moreover, anosmia may often follow head trauma as a result of damage to the olfactory cortex or due to the shearing of the olfactory nerves as they pass the cribriform plate. Now, anosmia is an abnormality with a number of disadvantages. For example, a person would be unable to experience enjoyment of pleasant aromas and also a full spectrum of tastes, which would suppress the appetite, and it could cause weight loss, depression, and withdrawal from social events that involve food. But the more alarming disadvantage is that the individual is at greater risk because they are not able to detect odor from dangers such as gas leak or fire, not to mention spoiled food. Then other two abnormalities are hyposmia and hyperosmia. Hyposmia is the reduced ability to recognize and to detect any odor. In this condition, the odors can only be detected at higher concentrations. Now, again, hyposmia, it may be temporary or permanent, and it could occur due to the same causes of anosmia. It is the most common disorder of smell during aging and even as a result of upper respiratory tract infection. Now, hyperosmia is the exaggerated sensation, and it is also called olfactory hyperesthesia. Interestingly, it is only a perceptual disorder, but it may occur in brain injury, even in epilepsy, and some neurotic conditions also. Since we are discussing olfactory abnormalities, so let's talk about the weird ones also. This phantosmia is just an olfactory hallucination. It means smelling something that's not there. Now, the disorder could be central or peripheral. Phantom smells, or you can say imaginary odors, they are not uncommon. And brief episodes of phantosmia can also be triggered by temporal lobe seizures, epilepsy, head trauma, or even at the onset of a migraine. Now, pardon me, I'm not comparing you with this superb animal, but do you ever wonder that why do we sniff to smell something better? Because it increases our ability to smell by enhancing the detection of odorous molecules in the air. Not only this, but sniffing also causes a peripheral drive in brain to synchronize the rhythmic activity, which is the concurrent firing of neurons in the olfactory bulb with breathing.